We will look at the 3:1 Punnett square ratio later, but first we will study the simpler 1:1 ratio because it is easier to understand. The 1:1 ratio occurs in genetic inheritance when a heterozygous individual is crossed with a homozygous recessive individual. This type of cross is called a test cross and is used to find out the genotype of an organism that shows a dominant trait. In this cross, the heterozygous parent produces two types of gametes in equal amounts, one carrying the dominant allele and one carrying the recessive allele. The homozygous recessive parent produces only one type of gamete, carrying the recessive allele. When fertilization occurs, the possible offspring are individuals that are heterozygous and individuals that are homozygous recessive. The heterozygous offspring show the dominant trait, while the homozygous recessive offspring show the recessive trait. Since both outcomes are equally likely, the offspring appear in a 1:1 ratio. This means half of the offspring show the dominant phenotype and half show the recessive phenotype. This ratio is useful because it helps scientists determine whether an organism showing a dominant trait is homozygous dominant or heterozygous. Now, here we are using the Punnett square method for an eye color determination with a 1:1 ratio. First, we write the parental genotypes. In this example, one parent is heterozygous with one dominant allele and one recessive allele, which is one capital B and one small b, while the other parent is homozygous recessive with two recessive alleles, that is small bb. Next, we identify the possible gametes. The heterozygous parent can produce two types of gametes, one carrying the dominant allele and one carrying the recessive allele. The homozygous recessive parent can produce only one type of gamete, carrying the recessive allele. After that, we draw a 2x2 Punnett square. We place one parent's gametes along the top, as both recessive represented by small bb are placed here and the other parent's gametes along the side, as heterozygous one with one capital B and one small b are placed on the side. Now we simply cross them and begin filling in the boxes by combining the alleles from each side. The dominant allele at the side that is capital B combines with the recessive allele at the top that is small b to form heterozygous offspring represented by capital B and small b, in the first box. Similarly, again this dominant allele that is capital B at side combines with the recessive allele at top represented by small b, to form heterozygous offspring represented by capital B and small b, in the second box. Next, the recessive allele at the side which is small b here, combines with the recessive allele at the top, also represented by small b, to form homozygous recessive offspring in the next box. Again, we do the same. The recessive allele at the side which is small b here, combines with another recessive allele at the top, also represented by small b, to form homozygous recessive offspring in the next box. When the table is complete, the outcomes show two heterozygous individuals with one capital B and one small b and two homozygous recessive individuals which are both represented by small b. This gives a genotypic ratio of one heterozygous to one homozygous recessive. In terms of phenotype, the heterozygous individuals show the dominant trait, such as brown eyes, while the homozygous recessive individuals show the recessive trait, such as blue eyes. Therefore, the phenotypic ratio is one brown eye to one blue eye,